World's first kidneys grown with human cells in pig embryos. Chinese scientists have succeeded in growing kidneys containing human cells in pig embryos, a world first that could one day help address organ donation shortages. But the development described in a study in the journal Cell Stem Cell on Thursday raises ethical issues, especially since some human cells were also found in the pig's brains, the experts said. The researchers from the Guangzhou Institutes of Biomedics and Health focuses on kidneys because they are one of the first organs to develop and the most commonly transplanted in human medicine. Rat organs have been produced in mice and mouse organs have been produced in rats, but previous attempts to grow human organs in pigs have not succeeded. Senior author Liang Shu Lai said in a statement, "Our approach improves the integration of human cells into recipient tissues and allows us to grow human organs in pigs. This is a different approach to the recent high-profile breakthroughs in the United States, where genetically modified pig kidneys and even a heart valve had been placed inside humans." The new paper describes pioneering steps in how a new approach in organ bioengineering using pigs as incubators for growing and cultivating human organs, said Dusko Illich, professor of stem cell science at King's College London, who was not involved in the research. Illich cautioned there would be many challenges to turning the experiment into a viable solution, but nevertheless, this captivating strategy warrants further exploration. Gene editing, a major challenge in creating such hybrids, has been that pig cells outcompete the human cells. To overcome the obstacles, the team used CRISPR gene editing to delete two genes essential for kidneys to form inside the pig embryo, creating what's called a niche. They then added specially prepared human pluripotent stem cells, cells that have the potential to develop into any cell type, which filled the niche, and before implanting the embryo in sows, they grew them in test tubes containing substances that nourishes both the human and pig cells. In total, they transferred 1,820 embryos into 13 surrogate mothers. The pregnancies were terminated at 25 and 28 days to assess how the experiment had worked. 5 embryos selected for analysis were found to have functioning normal kidneys for their stage of development, beginning to grow the ureters and would eventually connect them to the bladder. They contain between 50 and 60% human cells. The co-author, Zhang Dai, says, "We found that if you create a niche in the pig embryo, then the human cells naturally go into these spaces. We saw only very few human neural cells in the brain and spinal cord, and no human cells in the genital ridge. Preventing the invasion of human cells into reproductive tissue is considered crucial because otherwise there's a risk of uncontrolled creation of pig-human hybrids. But the presence of any human cells in the pig brain still raises concerns," said Darius Widera, professor of stem cell biology, University of Reading of the UK. He said, "Although this approach is a clear milestone and the first successful attempt to grow whole organs containing human cells in pigs, the proportion of human cells in the generated kidneys is still not high enough. In the long run, the team wants to optimize their technology for use in human transplantation, but admits it's not ready yet. An important limitation was the kidneys had pig-derived vascular cells, which could cause rejection if transplanted into a human. Nevertheless, the scientists plan to press on and allow the kidneys to develop for a longer duration. They're also working on growing other human organs in pigs, such as hearts and pancreas." This is by Agency France Presse on Science Alert. Please leave your comments and thank you for your support. Please support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support, and that you find all my content so interesting. You'll find the Patreon account details in the description box below.